Now we're ready to talk about synaptic transmission. So let's first go back and revisit our naked friend here. And so if you recall, he was walking around his bedroom and he stepped on a tack, and that pain signal, which was generated in a sensory neuron axon in his foot, was transmitted along the nerve axon by an action potential. So we talked all about how action potentials are produced and how they propagate. Later we'll talk about the sensory systems and pain in more detail, but for now we know that the way it gets to the spinal cord is through the action potential. The action potential travels into the spinal cord through those dorsal roots, and then it gets back to the signal gets back to the leg to create the motor reflex through a motor neuron that sends an action potential back down the leg to the muscle. But the question is, how does it get from one to the other? And so you can see here that in a typical reflex like this, you have the sensory neuron connected to an interneuron. So this is the interneuron, this is the motor neuron, this is the sensory neuron. And the places where they're connected are the synapses. So the synapses are these connection points. There's also a synapse here between the motor neuron axon and the muscle. So we know how action potentials travel. We know how they're able to travel so quickly, but the action potential can only travel, by definition, along the axon. Remember, and we talked about that, why that is. So the membrane of the axon is covered in voltage-gated sodium channels. We call it excitable membrane, or covered in myelin. But the action potential of the electrical signal cannot leave the cell. So that means it can't jump from the sensory neuron to the interneuron, or from the interneuron to the motor neuron, or from the motor neuron to the muscle. So there's no electrical signal in most cases that can cross the gap between those cells, which we call the synapse. So this chapter we will talk about how, the last chapter we talked about how information travels within a neuron, so mainly the action potential. This time we will talk about how information travels between neurons across synapses. So we'll be talking about synapses in this chapter. Mostly we'll be talking about one type of synapse, but there are actually two major types of synapses, the electrical synapses and the chemical synapses. Mostly we'll be talking about chemical synapses because chemical synapses are much more common, and they are much more important for most of the communication that happens within, especially in the brain and spinal cord. Electrical synapses don't show up really, at least in the mammal brain or the vertebrate brain, and they're really only significant in certain peripheral nervous system circuits. But we will talk about them first because electrical synapses are interesting for several reasons. First of all, if you remember, we talked about the neuron doctrine. We said that Camille Golgi originally proposed what's called the reticular theory, and then Santiago Ramon y Cajal came up with the neuron doctrine, which says that neurons are discrete and they are connected at synapses, but they don't share a continuous internal system. And Golgi said, he proposed the reticular theory, which says that neurons are connected to each other in a continuous network, kind of like the circulatory system. And what we now know is that, in a way, they were both kind of right, because what Ramon y Cajal was proposing turns out, as we will see, to be the chemical synapses, so gaps between cells that in places where they don't actually connect with each other. And their cytoplasm is not continuous. But electrical synapses, it turns out, are, in retrospect, kind of what Golgi was thinking of. Because an electrical synapse consists of these structures called gap junctions. And essentially, it's still a place where you have two separate cells. So we have cell one or neuron one, and these can exist between other types of cells too, not just neurons. But you have two cells that get very, very close to each other, and even though the membranes aren't continuous, they have these structures called gap junctions, and each gap junction consists of these proteins called connexins. and the connexins form these larger complexes called connexons. And then there's one set of connexon in each cell, and they connect to each other to form the gap junction. So this whole thing is a gap junction. So an electrical synapse consists of a whole bunch of these gap junctions connecting the two cells. And what's important is that each connexon is this sort of barrel-shaped channel, and the channel is connected from one side of the gap junction to another. So what that means is that stuff, water, and actually pretty much anything can go through these channels from one cell to the other. These aren't like ion channels that are selective. They're not even like the water channels in the membrane. They're called aquaporins. These are basically just big holes that almost anything can go through. So pretty much any kind of ion, water, other small molecules, even relatively big molecules like amino acids or small sugar molecules can easily go through these channels. And so what that means is effectively the two cells, for all intents and purposes, have the same cytoplasm. So the cytoplasm or the cytosol of each cell can go back and forth. And that's also true of charge. So any change in membrane potential due to current going in or out of one of the cells will have the same effect, or the current will just basically instantaneously move from one cell to the other. So if one cell becomes depolarized or hyperpolarized, the other cell will do the same thing. So if, for example, you have this situation where we're recording the membrane potential of cell 1, if you see an action potential, so this curve is an action potential occurring in cell 1, and almost at the exact same time, you will see a change in the membrane potential of the postsynaptic cell. This is called, that's what we mean by PSP. So this is the electrical postsynaptic potential. And because that's what we're recording here, is the membrane potential of this cell 2. So what that means is they effectively have the same membrane potential. It's a little bit weaker in the second cell because some of that current does leak out, but they're essentially linked up or synced together in terms of their membrane potential. And importantly, this current flow can go in either direction. So if it were the other way around, if the action potential occurred in cell 2, it would carry over into cell 1. So with electrical synapses, like I said, they basically have the same exact membrane potential, and for all intents and purposes, you can almost treat them as one cell, at least in terms of their electrical properties. So these electrical synapses, like I said, they don't show up very often, and in fact, we're not really going to talk about them much after this. They are very, very fast, so if there's an action potential in one neuron, it gets carried to the second neuron almost instantaneously, and they're bidirectional. So again, you can have signals going in either direction. However, as we'll see in a minute, they're not quite as useful as chemical synapses, mostly because there's only one kind of, you can only have one kind of signal. So whatever one cell does, it just gets copied essentially in the second cell. So there's no way for there to be a more complex signal to travel from one cell to the other. For that, you need chemical synapses. So that's why we're going to spend a lot more time talking about chemical synapses. So first of all, how do we know that chemical synapses even exist? Again, Golgi proposed the reticular system because, in a way, it's simpler. It makes more sense that you would just have signals directly traveling from one cell to the other. And it seems a bit complicated to have this gap where chemicals have to cross from one side to the other, from one neuron to the other. And really, Ramon y Cajal only really proposed the neuron doctrine based on just looking at the cells. So he had no direct evidence that chemical synapses exist or that cells weren't contiguous with each other. It wasn't until later that you had experiments showing that chemical synapses are in fact are real and are the main way that neurons communicate with each other and with other types of cells. So there are lots of experiments that have established that chemical synapses exist. One of the first and sort of most famous is this one carried out by this German guy, Otto Louis, in 1921. So what Louis did is he took the heart out of a frog, and but left the nerves intact. So this is a heart, and the nerve that controls heart rate is called the vagus nerve. So this is the vagus nerve here, and it's connected to the heart. It's connected to lots of other things, but the vagus nerve, if you electrically stimulate it, so if you attach an electrode to the vagus nerve and stimulate it, you will actually slow the heart rate. So this was already known, this has been known for a long time, that the vagus nerve, when it's active, causes the heart rate to slow down. The vagus nerve, and we'll learn more about this more later, is actually part of what's called the autonomic nervous system. And so again, Louis did this with a heart that had been taken out of a frog with the nerve still attached. But what he did was he put that heart in basically a little water bath, and or a fluid bath that kept the cells alive and allowed the heart to keep beating. And when he electrically stimulated the vagus nerve, at the same time, he removed a small amount of fluid from around the heart. And then, presumably then, if any chemicals were released by the nerve, it would be picked up by his little pipette here. And then he took that fluid and applied it to another heart, or the heart of another frog, that this time had the vagus nerve removed. So for this heart, the only thing controlling it is just its intrinsic electrical activity. There's no input from the vagus nerve. And importantly, the heart will beat on its own without any input from the vagus nerve. The vagus nerve comes from the brain, but all it does is control heart rate. It doesn't actually cause the heart to beat. So the heart will beat on its own with no nerve attached at all. But when Louis did this with the nerve removed and took that fluid that he'd gotten from the first heart and applied it to the second heart, he got the exact same result. So the heart rate slowed even though there was no nerve attached. And so the only explanation for this is that there must be something in that fluid that was being released by the vagus nerve that was causing the heart rate to slow. So that means that the electrical signal, in other words, the action potential, must somehow be converted or transmitted as a chemical signal from the nerve to the heart. Now, of course, this is a specific kind of synapse, but many other experiments have established that this is exactly how most synapses in both the peripheral and the central nervous system work. One neuron releases a chemical that causes the cell on the other side to react in some way. And so this is what we mean by chemical synapses. So we've talked a little bit about chemical synapses already. Typically, a typical chemical synapse looks like this in the brain, at least, in the central nervous system. So the presynaptic element of a synapse is really the axon terminal. So the axon terminal is just the end of an axon collateral. So this is the axon, and it's coming from a cell body of a neuron, and it ends or terminates in, again, here we're calling it the presynaptic element. Sometimes it's called the presynaptic terminal. And so inside the presynaptic terminal, you have mitochondria, but importantly, you also have these little bubbles of membrane called synaptic vesicles. So these vesicles are just little spheres of membrane, and some of them can come from the axon, so they're carried down the axon from the cell body along these microtubules. Some are produced in the synaptic terminal, especially these bigger, especially the smaller ones. The bigger ones, these bigger ones that we call secretory granules, come from the cell body, whereas these smaller ones that are sometimes just called synaptic vesicles are made in the synaptic terminal itself. And inside those vesicles and inside the secretory granules are the neurotransmitters, the chemicals that are going to be released. And then when they're released, they cross this little gap here called the synaptic cleft. So that's what we mean by just the empty space in between the presynaptic terminal and the postsynaptic membrane. So on the other side of the synapse, you have something. Could be a neuron, could be a muscle. If you're in the peripheral nervous system, there are even some glands and other kinds of cells that can be the postsynaptic target of a neuron or a synapse. In this case, it's a dendrite. So this is just the postsynaptic membrane of that dendrite. It could be anywhere along the dendrite, could be on a spine or on the shaft. Doesn't really matter. It does matter in terms of how the synapse works, but but on the postsynaptic side, they can be anywhere. And the postsynaptic side is kind of specialized, but importantly, what's on the postsynaptic membrane are these proteins called receptors. And these are the proteins that are going to bind to the neurotransmitter and then cause the postsynaptic cell to do something. And of course, this is just an artist's rendition of what a synapse looks like, but you can actually see synapses. So this is over here what a synapse looks like in an electron microscope image. And so everything on this side is the presynaptic cell. So this is the membrane of the presynaptic cell, and that teeny tiny gap, you can barely see the space in between, is the synaptic cleft, because on the other side, the bottom of this picture, is the postsynaptic cell. Now, the entire thing is not a synapse. In fact, the only place where neurotransmitters are released are at these regions that are kind of densely stained called the active zone. So the active zone is a place where the vesicles, you can see the vesicles here, these little balls, are kind of congregated, and it's very densely pigmented here because the stain that you use for electron microscopy, there are these metals that tend to precipitate around, especially proteins. So all the proteins involved in synaptic transmission are kind of collected at very dense concentrations at that place where the vesicles are going to be released. So we call that the active zone. And so in a sense, each active zone is like the fundamental unit of a synapse. So a synapse can have multiple active zones, and in a way, each active zone almost acts like its own little synapse within the larger synapse. Again, there's two kinds of vesicles. The little ones that in these pictures look almost hollow are called just synaptic vesicles usually. And then the bigger ones that also are very densely stained or pigmented are called dense core vesicles, or sometimes they're called secretory granules. But they're different in that they contain mostly what are called neuropeptides. So these are neurotransmitters that are actually proteins. We'll talk about them more in a minute. Whereas the smaller ones that look like they're hollow have smaller molecule neurotransmitters. And they are different in terms of how they're made and how they're released, and we'll talk about them, both kinds, more later on. So there are lots and lots of different types of synapses, and we can classify them different ways. So one way is by what part of a neuron they're connected to. So in the central nervous system, again, the presynaptic terminal is always the end of an axon, and the postsynaptic membrane is going to be on some part of another neuron. So usually when we talk about synapses, we think of the dendrites as being the antenna, or sort of the receiving area of the synapse, and that's where most synapses probably exist. And so if you have a synapse between an axon and a dendrite, we call it an axodendritic synapse. But you can have a synapse where the presynaptic terminal connects to the cell body of another neuron. Remember, soma is the other name for a cell body, so we call that an axosomatic synapse. Meanwhile, you can even have a synapse between, or where the presynaptic terminal is the end of an axon, and then the postsynaptic membrane is part of a different axon. So usually somewhere along the shaft of the axon, and so we call that an axoaxonic synapse. So that's just where you have synapse between an axon and another axon. And there's even more complicated situations. You could have, there are even neurons that synapse with themselves. So an axon can branch and turn around and connect with its own cell body or its own dendrites. You can also talk about synapses. It's important to point out that synapses come in all shapes and sizes. So usually when you see drawings of synapses, they look kind of like these guys, where you have a presynaptic terminal, a single presynaptic terminal with a single active zone, a single dendritic spine, and the synapse is on the sort of tip or the apex of that spine. But you can also have synapses where the presynaptic terminal is an axon that branches and actually forms multiple active zones with the postsynaptic cell. Here's one where the presynaptic terminal kind of wraps around the shaft of a dendrite or maybe the cell body and forms many different active zones. So each one of these little active zones, again, kind of acts like its own separate synapse. Or you could have a situation like this where the postsynaptic elements are, again, maybe little dendritic spines or maybe neurites from other cells. And the presynaptic element is one big giant bulbous mass that again has a bunch of postsynaptic or presynaptic active zones on it. So the point is that there's no single way to describe synapses. Again, for convenience sake, a lot of times they're drawn like this simple one-to-one relationship, but in reality, most synapses are much more complicated looking. Another way to describe synapses is in terms of their symmetry, which means that, again, if you look at the membrane of the cell, so again, you can see that best if we go back to these pictures. The active zone has this sort of density on either side. There's a presynaptic density and a postsynaptic density. And if those two densities are different, meaning if one is thicker than the other, especially the postsynaptic density, we call that an asymmetrical synapse. Whereas if the pre and postsynaptic densities look about the same in terms of thickness, we call it a symmetrical synapse. It turns out that there's a functional difference between those two. So the asymmetrical synapses are sometimes called Gray's type 1 synapse, and the symmetrical ones are called Gray's type 2. Typically, the Gray's type 1 synapses are what we call excitatory. And so that means that when those synapses are active, the postsynaptic cell experiences depolarization and then usually an action potential. We'll talk about how that works in a minute. And then the type 2 synapses are typically inhibitory, which means that they actually reduce the electrical activity or reduce the chances of an action potential in the postsynaptic cell. And then, of course, you can have synapses between neurons and other types of cells. So out in the peripheral nervous system, you can have synapses with certain glandular cells, but especially with muscles. So the way that the brain controls your body is by causing muscles to contract. And we'll talk all about muscles and muscle contraction and the motor systems in a later chapter. But the synapse between a neuron and a muscle is called the neuromuscular junction. And in fact, this particular kind of neuromuscular junction is between what's called a motor neuron. So a motor neuron is just a neuron in the central nervous system, mostly in the spinal cord, although there are some in the brainstem too. It has a long axon that goes all the way out through the peripheral nervous system to these fibers of muscle, especially what's called skeletal muscle. So skeletal muscle, as we'll learn later, is the muscle that you actually use to move your body. And the synapse that they form is called the neuromuscular junction. So this whole thing is a neuromuscular junction. It's a little bit different from the central nervous system in terms of in some properties, but it's exactly the same in others. So it still contains synaptic vesicles, and those synaptic vesicles are released from the synaptic cleft to release neurotransmitter, which then binds to receptors on the postsynaptic side. We'll also learn later that the neurotransmitter that gets released from those vesicles is always called acetylcholine. So in the central nervous system, you can have lots of different kinds of neurotransmitters, but at the neuromuscular junction, it's always acetylcholine. And so on the postsynaptic side, you have acetylcholine receptors. We'll talk about those more later too. Structurally, though, the neuromuscular junction is different from a central nervous system synapse. For one thing, it's much bigger. So the surface area of the synapse and the area of each active zone is a lot bigger than a typical synapse in the brain. And so each axon usually branches and forms lots of little terminals. So each of these is a separate terminal, and at each position here, you have, if you look underneath, you'll see this whole area, which is called the motor end plate, is covered in these little folds called junctional folds. So this is kind of a cross section through the membrane, and each of these little grooves is part of that motor end plate area. And what's important is that those grooves create a lot more surface area, which means there's a lot more chances for neurotransmitter to bind and to actually activate this muscle. And that's the functional difference between neuromuscular junction and a CNS synapse, is that they're faster, so the response from the muscle to neurotransmitter release is much quicker. Again, that's because of just the amount of neurotransmitter being released and the number of receptors that are available to bind. And they're much more reliable, which means that a given action potential from the motor neuron is guaranteed to cause the muscle fiber to react, to respond. which is not always the case with central nervous system synapses. We'll talk about that later. But not all action potentials in a central nervous system neuron are going to trigger action potentials in a postsynaptic cell. And that actually is important because that's how the central nervous system is able to process information. So we'll come back and again, we'll talk about muscles more later. But for now, we're going to stick with central nervous system synapses. And so next time, we will talk about neurotransmitters themselves.